right. So let's continue. So right now we're gonna switch to a couple of techniques that essentially are for more exploratory analysis and looking at the data from a more holistic perspective and try to identify what's going on. And we have already seen a number of examples in numerous presentations based on principal component analysis or cluster analysis. So again, I'm not gonna get into all the details, but just give you a flavor of what is going on and go back to some of the plots and pictures that we have seen and make a few comments. So in many presentations, some speakers essentially use the two techniques a little bit interchangeably. And on the other hand, there are some key differences, and I would like sort of to make that distinction of exactly what the techniques are designed and when they give you similar results and when you may want to use one over the other, depending on what questions you're interested in. So principal component analysis is a very old technique, almost 90 years old. And the idea is essentially you have this high dimensional cloud because you have a number of samples living in 450 dimensional space, given the fact that you have 450 lipids, and essentially you would like to see what is going on or what is interesting in your data set. So essentially you're trying sort of to figure out directions of maximum variability. I'm going to show you a picture on the next slide in this high dimensional cloud. And principal component analysis leverages correlations in the data. That's not true with cluster analysis. So that's one of the key differences. And these directions essentially are linear combinations of the original features, the metabolites, and therefore in many cases they have a nice interpretation. Because you may find that, for example, one of your principal components but define these directions corresponds, let's say, to the amino acids, and therefore that is its interpretation, and that's what sort of TM did yesterday with one of his examples. So this is exactly what I mean. So this is a toy example. I have, let's say, two metabolites. They're highly correlated. So essentially, the only interesting thing that's going in this data set is if I were sort of to look at the data from this perspective. There is nothing going on in the other sort of direction, which means that if I project it and essentially think that I projected all the data and they just sit on the line, essentially I see what's going on. I can identify some outliers and so forth. And the higher the correlation, essentially there is nothing left sort of to explore. So if you take this picture and somehow magically your 450 featured data set, because it has 450 lipids, could be mapped to a single line, essentially you can tell a lot about what's going on in your data if that was a successful exercise. Which means that essentially it implies that if all your features were very highly correlated. So for example, principal component analysis is perfectly suited for lipidomics data sets because we know that there is very, very strong correlations within the various lipid classes. So this is a good way to explore the data sets. Similarly, to some extent, to the metabolomics data sets, especially for particular sets of metabolites. But the key aspect is whatever you get from principal component analysis because you have strong correlations, and that's what allows you to summarize the data successfully. So just to show you a few pictures from the PUFA study. So for example, this is a Pca plot. I'm using 3 principal components, and you can see sort of various patterns. You see this fairly tight cluster, and then a few other patients or subjects floating around. This is another plot where I'm looking at, I'm labeling the points. If you see, apart from this slight rotation of the 3D picture, essentially I have exactly the same points. I'm just coloring by different metadata, the visits, or the 2 conditions, control versus FLD, and so forth. And I'm trying to see sort of whether essentially patients or subjects in one condition naturally fall within a nice cluster and so forth. Although principal component analysis is not designed to find clusters. It leverages these correlations, and you should never forget that. There is an important concept when you look at the results of principal component analysis, and this is sort of important because as part of the report, you also get a Pca plot of your samples. So this is the concept of total variance explained, and most of the software, essentially they give you such a number along the principal axis. So for example, in this case, it tells you that 43% of the total variance was explained by the first component and another 10% by the second component. So essentially, you see that by just looking at this picture, you have accounted for something like 55% of the total variability. So essentially, you have a very nice summary of your data, and you can sort of start exploring what goes on. The most striking, if I were thinking about this plot from a quality perspective, quality control point of view, I see these three outliers. And principal component analysis, and I'm going to show you another example, is pretty good technique to identify potential multivariate outliers. And you may start questioning and then go back and interrogate, these 3 points, whether there was something special going on, or there was a problem, and so on and so forth. Or potentially, these are maybe the most interesting samples that you have in your study. So essentially, the total variance in the data set is if you sum the variances of all your features or all your metabolites, and then you're trying to figure out these combinations, weighted sums of your original features, how much of this variance explained. So if somebody gives you a Pca plot and here you have 5% and there you have 3%, just throw it away. Essentially, you haven't learned anything from principal component analysis, because that means that there is more than 90% of variance unexplained. So by looking at this picture, you're overinterpreting. There is too much activity that is not captured by this picture, and therefore is completely useless. The moment these numbers start adding up to something over 50%, in some cases going to a 3-dimensional plot like here, then you have captured enough of the variability in your data, and then you can start exploring the patterns and see if anything interesting occurs in terms of potential outliers, or you see sort of nice clusters and so on and so forth. So this is an important statistic or an important summary measure that you need to look at. If this number is tiny and this number is tiny, stop there. Essentially, principal component analysis doesn't provide any insight into the data set, and you're essentially wasting your time. So some applications of principal component analysis: quality control purposes, identify multivariate outliers, we already saw an example, and essentially obtain a summary view of the data like the plots that we saw. So this is a picture that I borrowed from TM slide. So essentially, the first question that, given the fact that he was developing the lipidomics platform and he ran his 4 cell lines and at different time points and so forth, essentially you would like to see whether the replicates go together. And the reason is that, given the fact that these are cell cultures, the results should be highly reproducible, and therefore within the samples coming from the same cell line, they should be very highly correlated. And given the fact that Pca leverages correlations, you better have these very, very tiny clusters. So as an example, suppose that one of his link ups, so he had 3 replicates, 2 were going together and the third was going there, automatically that raises a red flag. It means that one of the replicates had technical issues, either at sample preparation, extraction, something went wrong at runtime, and so forth. So for the core, but also for you, by having such a plot, once, of course, you look at this percentage of variance explained, so that the picture is representative of what goes on in the data, is great for quality control. So essentially what this tells TM is that his data are of supreme quality, this superb quality, and therefore then he can sort of proceed and start doing analysis. The other thing that you would like to see, or the core would like to see, is remember that both Maureen and TM emphasize the role of test pools. Essentially, you take material from all your samples, you pull them together, and then we run them. These need to be somewhere in the center, because essentially this is a combination of material from all your cell lines, and this is another validation that the quality of the data is pretty high. So especially with animal studies or cell culture studies, PCA, because you expect that the replicates should be highly reproducible, it provides you a lot of confidence about the reproducibility of your results, and then you can move on and start doing the more interesting and high-powered statistical analysis. Here, we've seen a previous picture. This point is a red flag. It may be a very interesting point from a biological perspective, or something went terribly wrong during the processing of the data. This is another picture from Chuck's slides. Again, here you can use it both. What are the blues? The pools. So here you're looking at a more complicated picture. So the first thing is that the pools better cluster very well. If, for example, the pools were all over the place, again, going back to quality control purposes, then you get skeptical about the quality of the run. But then in this case, given the fact that we have different samples, then you're trying essentially to see whether there are interesting patterns in the data. And again, in this particular case, principal component analysis serves as a way of summarizing these high-dimensional data sets and see whether the results are consistent or you start noticing something interesting or alarming. But the key aspect to remember is principal component analysis leverages correlations. And especially for the lipidomics platform, as I've already mentioned, given the fact that lipids class are highly correlated, this is a great technique sort of to explore your data. Jack, it's your plot.
The problem with the graph is that these are totally different studies that were just run all at the same time, and then all the data was compiled. And so it's hard to say what's going on when you actually have different kinds of studies with different individuals that cause homogeneity. But if you look at it, you can see that if you look at the red overfeeding versus the green boxes versus the red triangles, they separate. And so the overfeeding of 2,000 calories a day will change the lipidome, not surprisingly. But now we can actually see which ones are driving. If you look, you're going to talk about loading and all that kind of stuff. But now we can see the unsaturated, saturated fatty acids. If you actually start looking at protein, you can actually see these things separate. It was just more to, it's actually the service that Anu provides, which does the analysis, to do a Pca plot, because she's agnostic to what the groups are. So anyway, but what's important to me is what are the lipids that will signal that if you're in a positive energy balance, what are the lipids that are going to change? That's what I wanted to know. There's actually somebody else did this study for a completely different reason, and I got the blood to be able to do that.
So we would look at that plot to see separation by quadrants or by either axis, or
I look at the distance, the sort of the circle around and look at the distance, there. The PUPA data is actually look at, I would say you can't see it, probably, I don't know if you're going to, yeah.
Not really.
Go back
Yeah.
to this one you can't see, but the problem is that there's a number of data points, time points on here. But
Yeah.
one plot that we create from this, you can see that as you feed the high concentrated peptide, they move in a temporal manner a little bit differently in the high, if people not out lost any liver disease, things like that. So those are the kinds of things you see. It's not the best example to look at. Later I can show you something. In fact, I'll give talk this afternoon to show you how you can use it.
So going back to TM's slide, I emphasize, from my perspective, how you can use for quality control purposes. But the other thing that you can sort of elaborate on what Chuck said is, you see that, you know, the link up cell line is further away, and it's, so on the first principal component, which is defined along this PC1, essentially the V cap and the link cap are about the same, because essentially they hug the zero axis. But on the other hand, they are start being separated on the second principal component. So the output from the technique, and unfortunately I don't have such a detailed output, starts telling you, you know, remember, the technique, essentially what it does, takes these weighted combinations of all the metabolites in the data set. So essentially what happens in this plot, for on the second principal components, there are a certain number of lipids that come with a negative sign on the, as the weights in this combination, and then there are another set of lipids that come with positive weights, and essentially then that's where you start sort of digging in a bit deeper and see which of the lipids start differentiating between the V cap and the link cap. The other thing that you see is that based on this plot, and provided that the amount of variance explained by this three-dimensional plot is adequate, so this is a meaningful plot, the RWP cell line is fairly close to the PC cell lines, and so on and so forth. And therefore, remember that some of these cell lines are androgen dependent, and some of them are androgen independent, and you would like to see whether the androgen independent are closer together, and the androgen independent are further away from the androgen dependent, and so on and so forth. So once you have some information, so here primarily I want you to emphasize the quality control aspects of the plot, but once you start having information about your samples and potential groups, this is what you would like sort of to see and potentially conclude. Another way, as Chuck mentioned, in many cases, if you have data from two different studies and you would like to see how concordance there are, then you run a joint PCA and you would like sort of to see if samples align.
Can I ask?
Sure.
As the PCA analysis shows these differences among different samples along various axes, does the report reflect those differences? So, because if the report is just basically an Excel sheet with fold changes, it will be harder for the end user to tease out those differences.
So,
does the report show those differences reflected by the PCA analysis?
Tanu? So the question is, so suppose that you get this picture and of course you have the raw data, how can you tease all these differences reflected in the PCA plot? Yeah, but if you would, yes, but if you want to dig deeper, then we go back to the things that we were discussing before the break. Then you need to start running all these t-tests in order to really figure out what is differential, you know, which lipids really differentiate. The key advantage of a PCA or a cluster analysis, we'll come back to that in a second, is that instead of start doing 450 comparisons and taking care of all this business, it gives you in one single plot, if you have explained enough of the variance, a bird's eye view of your data set, you know.
George, Tanu is going to show PCA in some detail in the hands-on session, so you will be able to see exactly what she was talking about.
All the workings and all the fixings. So here was another kind of a fun application. So we have data. This was a ring trial, the infamous ring trial that NIH suggested to the MRCs. And essentially there are 50 samples, 20 from males, 20 from females, and 10 pools of the other 40 samples. And the question was, without knowing out of these 50 samples who is who, just running a metabolomics or lipidomics platform, can you identify the groups? And of course, the way I posed so this was the question that it was posed, and my working hypothesis, of course, you know, if you run a steroid, if you look at steroids, you can easily tell the males from the females. But to me, it was kind of an interesting challenge to think about the platform. These are not from the MRC, these are from Florida data. And essentially, my working hypothesis was, but the compounds, this is LCMS untargeted platform, that the compounds that exhibit high variability, and therefore these are the ones that contribute mostly to PCA, are indicative of the group differences. So essentially, so here I'm not even looking at, I don't know any of the compounds. I just got the Excel spreadsheet, I just selected compounds of high variability, and then I just dump them into a Pca Plot, and I know that in this picture I have explained 56% of the total variance. So this is an informative plot, and what you can see is one group there, another group there, and in this particular run, the pools, because they are sort of a combination of the material from the males and the females, are somewhere in the middle, although the pools were not sort of very tight. Maybe that was a problem with the run, of the sample preparation, and so forth. But to me, it was very interesting that, using this very naive or dumb strategy, you can easily sort of tell them apart. Of course, what I cannot tell apart without taking a further look at the compounds is whether these are the males and these are the females. But, this is kind of a fun application of PCA, because again, there is a lot of information based on these correlations of how you separate the groups. If you really are interested in groups, this is a nice segue to cluster analysis. If you really want to identify groups, then PCA, in many cases, it works well. But if you are really after groups and tell you what is going on, then you need sort of to get into the business of cluster analysis. And we're going to see Chuck has a demo of CoolMap, and I think there is a session tomorrow with a more hands-on. So the idea of cluster analysis is essentially you look at the data and you're trying sort of to organize them into homogeneous groups. So at the technical level, the key aspect is what do you mean by homogeneous groups? And the literature is literally enormous. If you put cluster analysis algorithms into Google search, it spits out maybe 15,000 papers. So everybody and his brother has tried to come up with a better clustering algorithm. It has a very long history. There are lots of algorithms. Many of them are implemented in various platforms. And essentially, you're trying sort of to look at similarities between either the compounds or the subjects or the experimental conditions and come up with this type of groups. Here I have kind of a menu of various classes of algorithms, and most of them you're going to find them in a lot of tools. For example, metabo-analysis, all these hierarchical methods are implemented, and also partition methods. There are more fancy ones that are based on statistical mixture models. The latest and personally to me the most powerful generation are these graph partition algorithms that take ideas from graph theory. Some of these, for technical reasons, they are a little bit more fancy and depend on initialization parameters. Something that came out of the TCGA project, was this consensus clustering idea, and I'm going to briefly discuss it in the afternoon in one application of integration of omics data. Generally, it's a very rich literature, and you have a lot of choices. So what I'm showing you here is how you can combine cluster analysis with differential analysis. So essentially what I'm doing here is there was a targeted assay. There were two conditions. Actually, these are some data from NASA. The C's are the mice that stayed on Earth. The F's are the mice that went to space. And essentially what you're trying to figure out is two things. Because here I'm doing cluster analysis two ways. I'm trying to cluster in one way the samples and see whether they sort of the mice that went to space and the mice that stayed on Earth cluster sort of together, and I see that 4 of those are fairly homogeneous, and some here definitely I spot an outlier because this looks like, I don't know, these mice went crazy. I thought that it was in the wrong environment. But also, if you do the cluster analysis on the metabolites, you are trying sort of to see, how the metabolites are similar and correlated with each other. And then, of course, at some point, this is not a very clean picture. So the next step that you do is you run your favorite test, here a t-test, and therefore, since you have two conditions, you only keep the metabolites that strongly differentiate between the two conditions. C7 is still goes with you know, the mice that went to space. And here, of course, I did the adjustment of the p-values. And essentially what you start seeing is a lot of oxidative stress and so forth. So this is an example of, you know, you're trying to find the groups. There are two ways of using the analysis, either on your samples or on your metabolites to try to understand what goes on. But in most cases, the most successful application of cluster analysis is you combine it with differential analysis that we discussed before the break, so that, you know, you clean up the picture and you see exactly what goes on. A few comments on cluster analysis versus principal component analysis. In many cases, people use them sort of interchangeably and try to look for groups, but the principal component analysis leverages the correlations. And essentially, you're trying to use it for summarizing your data. On the other hand, in cluster analysis, you explicitly are asking the questions, can I find homogeneous groups in my data? And in many cases, what happens is, yeah, you can find these subgroups. Yes, this is a very clean subgroup, and this is a fairly clean subgroup, and then you have some things that are in between. And in many cases, that's very informative because you say, okay, I'm going to treat this the same, and then in between, I have this much more heterogeneous group. So the goals are slightly different, and as I said on many occasions, the findings are similar, but this is not always the case. And something to keep in mind, both for principal component analysis and for cluster analysis, if you know the groups a priori, like in this particular case, the mice that flew to space and not, then you can apply differential analysis, essentially select important differentiating features, and then sort of clean up the analysis and the results. One important thing to remember both for principal component analysis and cluster analysis is there is no natural mechanism built into the procedure that selects the features. You know, here, when you start doing cluster analysis, essentially you put in all the metabolites available. So unless you do a filtering step, maybe through differential analysis, the technique doesn't have a built-in mechanism to select important features. So that's something to keep in mind. And in many cases, that's why when you use either principal component analysis or cluster analysis, you don't get very clean results, because many of these features, to some extent, create noise and don't give you clean patterns. So that's exactly what TM did with his cell lines. You know, after essentially he filtered the most important and differential lipids for his cell lines, essentially he gets this very nice and interpretable clustergram where he can sort of both tell the four cell lines apart, but also he has this nice clustering in terms of the androgen-dependent versus androgen-independent cell lines, which is biologically meaningful and so forth. So filtering always sort of helps, because essentially you throw out features that, or lipids or metabolites, but are not sort of discriminating between conditions of interest. So switching gears, predictive modeling. What are the biological questions of interest? Either identify a biomarker panel for distinguishing between controlled treatments. Think of whether you would like to identify the control subjects versus the NFLD subjects, and also validate the predictive ability. And this is a point that I'm going to come back later on. And this is a special class of predictive model called classification models. The other big class of such type of modeling is you would like to correlate some particular clinical outcome like survival times, non-recurrence, exposure maybe to some environmental factor to a particular biomarker panel. So at the high level, just to define a little bit of notation, you have an outcome variable of interest, either the survival time of patients or the level of environmental exposure or disease risk. And then you have a whole bunch of predictor variables. So the outcome variable is the y, and the predictor variables are x, and f is a function that is going to try to relate the two. And u is just a noise term. So what are the predictors? Well, the primary predictors, since we're in a metabolomics workshop, are the metabolome or the lipidome. But in many cases, you complement, as we discussed in epidemiological studies, with other background and clinical covariates. So let me go back to Farsad's presentation yesterday because it was a lot of technicalities. So let me sort of try to map them into this framework. So he was looking at cardiovascular disease risk, and essentially one of his outcome variables, the study had a number of outcome variables, was time to event. So, survival time in some sense, or until an event happened. And in terms of predictor variables, there was the lipidomic profiling, plus a lot of clinical and personal characteristics. And then there is a particular model, and in this case, it was this Cox proportional hazard model. If you want to find out more, this is a special model created if your outcome variable are survival times or time to events. If you have, for example, environmental exposure to some other factor, then this is not the appropriate model. Then you need to look at other regression models and so forth. So this is a very particular class, heavily covered in survival analysis biostatistics. However, in this particular case, and essentially something that Maureen brought up during her presentation about the untargeted platform, there is a big technical challenge. But the number of variables, given the fact that if you're doing untargeted profiling or lipids profiling, you have 450 variables. And in order to be able to run these models properly, and that's what I was taught in grad school, you need to have more samples than the number of variables. And this paradigm that probably you saw in a classical statistical course breaks down the moment you have more variables or more features than samples. So essentially, if these people really wanted to run without doing more fancy stuff, in order to do any of this modeling, you need to have humongous sample sizes. So that's a modern technical challenge, and the solution is a very new class of techniques developed in the statistical and machine learning literature the last 10 years. Farsad mentioned a number of times the word lasso, whatever that is, but essentially identify, even in this paradigm where you have more variables than samples, which is kind of a weird paradigm from a statistical perspective, they identify, based on some tuning parameter, the most strongly predictive variables. So if you would like to build such models, talk to an expert. This is very recent technology, and although you can do it yourself, maybe in R and stuff like that, there are a lot of very subtle issues of how you tune the procedure. So before you report the result, at least consult with a friend that has been in this business and knows what they're doing. Don't try this at home on your own. Maybe 5 years from now, all these things are going to be nicely packaged in software, but this is a very active research area. I'm one of the contributors, and I know a lot of the pitfalls. So this is something. It's very powerful because it allows you to start essentially correlating the whole lipidome or metabolome with particular outcome variables that you're interested in, but there are a number of technicalities. There is a special case where the outcome variable is binary, so disease status, yes or no, and then the predictor variables are similar. And one can use the previous modeling regression framework, and maybe many of you have heard about logistic regression. However, for this very particular setup, there is a full class of special models that have been developed in the literature. Again, and these are called classification models, and you have seen a lot of examples or you have heard all these terms during presentations: partial least squares, discriminant analysis, support vector machines, random forest, and so forth. So these are special techniques, but essentially trying to build a biomarker panel that can discriminate between two conditions. So the outcome variable is either binary or takes a selected set of values. It's not like a time to event or a continuous variable. The key aspect, in addition to keep in mind, is that the focus is on prediction rather than on gaining insight into biological mechanisms. Why am I saying this? Because some of these models, like support vector machines or partial least squares, are very, very complicated. They do the job in the sense that they have very high predictive accuracy, but they are almost black boxes. So there is a trade-off here. These may not be interpretable models. So this is the formal setting. You would like to build such a model where essentially F maps the predictors to the binary outcome so that essentially if then somebody comes with a new sample, you can classify it accurately. So for example, the metaboanalysis software has a number of options, including random forest, partially squares, discriminant analysis, support vector machines. So these have become very popular, very, very powerful methods. What I'm trying to show you is, I mean, this is kind of a toy example, how the different techniques separate the classes. So essentially here we had three classes, the O's, the triangles, and the pluses. And depending on how the technique, and essentially you would like sort of to draw these, what are called decision boundaries to separate the classes. So some techniques draw these linear decision boundaries. Some other type of techniques draw this quadratic type of decision boundaries. Some other techniques like support vector machines draw more fancy boundaries. Some techniques like k-nearest neighbors, essentially, you know, potentially they overfit and they draw very nonlinear boundaries. Decision trees draw this type of boundaries. The nice thing about decision trees is that it's very easy to interpret them because this is a very interpretable model. It tells you that if on this variable you're less than 24.5, you belong to this class. If you're larger than 24.5, well, then you need to look at another variable and then split you. So essentially, these models are very, very popular in medical informatics because they are very transparent. However, they're easy to interpret, and therefore, for example, if you were using such a classification tree, the biomarker panel will be easy to interpret and explain it to your colleagues. The downside is that they're not as powerful. There are all these more advanced methods that give you much higher predictive ability. So this is usually the trade-off that happens in life. You have something nice, simple, and interpretable, but it doesn't do as well as more complicated techniques. So what people then said is, instead of looking at a simple classification tree, let's create a whole bunch of such classification trees. The details of how you create those are in the appendix of my slides, and this is called a forest or a random forest. And therefore, through a complex procedure, the nice thing about random forest is they also tell you which of the features are important, although in this particular case, the final model, again, becomes hard to interpret. It becomes semi-black boxy. So this is a picture from Chuck's slides, and essentially in this particular case, the key variables that differentiate between the classes are this one. So essentially, this lipogenic index is the most important variable, this particular lipid, and so forth. The rest of the variables contribute a little bit in terms of the classificatory power, but essentially this is sort of how you use this type of techniques. So these classification models are a special class of predictive models where the outcome variable is of a binary nature, whether you have the disease or you don't have the disease, and so forth. So the take-home message is the overall statistical analysis driven by two main factors, the study design and, of course, the biological question of interest, namely whether you're an epidemiological study and you would like, for example, to relate metabolomics and lipidomics profiling to particular outcome variables, or whether you're trying to go after a biological mechanism in cancer or in diabetes and so forth. So just revisiting two examples, the prostate cell line study, it's a fairly simple design, 4 cell lines, and essentially the main steps of the analysis were a principal component analysis for assessing quality of the data, then you identify differential lipids either through a whole bunch of t-tests or an ANOVA model, and then followed by cluster analysis to visualize the results. On the other hand, in the PUFA study, as I've already emphasized, it's a multifactorial study. You have various conditions plus time course data. So, of course, you can do exploratory analysis to gain the bird's eye view of what's going on in your data, but then your main goal is to identify the differential metabolites, and then you need to build probably a more fancy statistical model because you have also the background information on these people to address heterogeneity issues, plus also you're interested potentially in building this classification model for predicting future patients. So if Chuck measures a patient, measures the lipidome using one of these random forest models or something similar, then you can predict by just looking at the lipidome whether he belongs to one of the classes. So the last thing, 2 slides only. We had a very brief discussion before the break about pathway enrichment analysis. A whole bunch of presentations in the afternoon focus on pathway enrichment analysis. So the key question, the biological question of interest is whether a set of functionally linked compounds is differentially expressed across 2 conditions. So right now you don't look one compound at a time, but you would like sort of to start taking a systems perspective. Essentially, you know how the compounds can be arranged to biochemical pathways, or they belong to particular lipids classes, and you don't care about any particular metabolite, but you would like sort of to start looking at them as a set. This is a great reference. Pathway enrichment analysis came out of people wanted sort of to look for enrichment of Go terms, and then a seminal paper was out of the Broad Institute in 2005, G-set enrichment analysis, and a lot of techniques have built on that type of work. By now we have 3 generations, and Nala is going to, in her slides, go a little bit more into those. The only thing that I would like sort of to point out is, again, some caveats or some things that you need to keep in mind. In gene-set enrichment analysis, essentially you have a deep coverage of the transcriptome. So you have a good representation of the pathways that you're interested in. In metabolomics, you may not have such good coverage. So that's something that you always need to keep in mind. The other thing that, of course, happens is that the pathway overlap. So there might be some common membership, and that induces some positive dependence. And therefore, when you get into testing issues, you may need to be a bit more fancy, and that's essentially the main point that I emphasize. So we're going to hear much more about pathway enrichment in the afternoon. In terms of tools, there is a presentation by Jeff on MetaboAnalyst. If you are good at scripting, the learning curve is very steep, but the R software, the R statistical language, and the associated package in the bioconductor are very, very good. Unfortunately, the learning curve is very, very steep, and a lot of it's essentially learning a software, a programming language, a high-level programming language, but essentially anything that you ever wanted to do to analyze omics data, this is a very full set of tools. There are some commercial tools, and some of the procedures that I mentioned are built in those. Chuck is going to show you CoolMap. I think there is a session on Medscape, a number of interrelated tools, essentially for pathway mapping, and then looking at the data from a pathway perspective, metabolic data from a pathway perspective that Ala and Bill are going to give you tomorrow. Questions before lunch?
Can I ask you to?
Sure.
So one issue that was sort of mentioned in experimental design and several times talked about was sort of, having your test data set which you use to train your model or build your model and having a validation cohort. Can you sort of comment a little bit on that? Because I think people are becoming increasingly aware of that, and I think sometimes it's sort of misinterpreted.
So this business comes up a lot in this context. Essentially, what Ala is referring to is, in order to build a lot of this type of models, one danger that you always run is you use a certain set of your samples, so you have the metabolic profile and other background variables and whatever outcome variable of interest. So this comes primarily here, in this business of predictive modeling. So you create this model, this fancy function f, And you have a certain type of results. Okay, you are saying that, yes, I can discriminate my classes, or I can predict very well the cardiovascular disease risk that Farsad showed you yesterday, that study, and so forth. And Simon's presentation this morning, again, had a lot of such advanced modeling. So one question is, whether, and there is always that danger, especially when you start getting to these very complicated statistical models, whether you are overfitting the data. Essentially, that the function f tries, and this is a good example. You see here the separating boundary tries to follow this particular data set very, very closely. On the other hand, here you have a straight line, so the straight lines are very robust. But in this particular case, potentially you're overfitting, because if the next time there is another plus observation somewhere here, you're going to sort of misclassify it. So how do you validate these very sophisticated models? So the best way to validate them is essentially to get a new data set, and therefore, so you build the f function from one set of your data, and then you bring an independent cohort where you plug in the x's to the previously trained f function, you spit out the predictions, and then you compare the predictions that you make out of your model with the truth on this independent cohort. If these start matching, it means that your f function is robust. Because the problem is that in this particular f, if you train and test your model on the same data set, potentially you run the risk of overfitting. Yes, there is a lot of technology and statistical literature of how to avoid overfitting and so forth, but the real proof of the pudding is when you bring an independent data set and you use it on a previously trained f function, and that's sort of the best way to go about it. The downside is, of course, that bringing a new independent cohort is expensive, how to collect the data and so forth. So that's why there is an extensive literature of using a number of technical approaches. Many of you may have heard the word cross-validation, where you keep reusing the same data set in a pseudo-independent manner. But the ultimate proof will be to bring a new cohort, but there is a price to pay. Essentially, you need to collect a new cohort. Yes.
So the random forest classification case, I think there should be a lot of samples, more than 200 samples. But in Chuck's experiments, the proof study only has 10 controls and 5 disease objects. So is it suitable to use this random forest? And then can we believe that the output for that two significant ones? How much we can believe that, the output?
So, this is a good question. I would disagree with the number that you gave about 200. I've run random forests on 50 or 70 samples. With a very limited number of samples, then definitely the model starts becoming too sensitive as you train it. Exactly, it's this, oops.
Overfitting problem.
Exactly, you start running into the overfitting problem. Again, if you have very strong features that essentially can really separate, the model is going to find them. On the other hand, if all the features contribute a little bit, then probably you need larger samples. In this business of statistics, everything is about signal to noise. If you have very strong signal and little noise, everything works, and you don't really need statisticians. You need statisticians and all this more fancy modeling when your signal is of adequate, but you have a lot of noise, and then that needs to be handled carefully.
And also the imbalance problem, the control and case numbers?
Yeah, in this case, it's not that bad because, essentially you have 2/3 in the control, 1/3 in the other group. But essentially what you're referring is, suppose that I have 100 samples and 15 belong to one group and 85 in the other group. So when you're trying to build this classificatory model, that becomes a very challenging setup because, you know, the distribution of samples across the two groups is not fairly balanced. You don't really need perfect balance, but when you start going into such extremes, life starts becoming very hard.
Thank you.
Any other questions? Time for a photo then.